For 50 years, the Nobel Conference at Gustavus Adolphus College has hosted preeminent scientists, theologians, and ethicists to discuss deep questions in the intersection of science and society. From the newest results in physics, chemistry, and biology to the newest fields of multidisciplinary study, scientists at the Nobel Conference have examined the universe at its largest and smallest scales, explored the oceans, and described new materials. Conference speakers have debated the mechanisms of aging, as well as science and economics of food. Often, speakers have given us a glimpse of the next big questions and how they might be answered. Throughout all of the conversations, ethicists and theologians have grounded the science in a human dimension. The 50th Nobel Conference at Gustavus is celebrating a half-century of bringing breakthrough science to lay audiences in the upper Midwest, across the country, and recently around the world. Nobel Conference 50 is assembling previous Nobel Conference participants to look at recent advances and future directions in the physical sciences, evolutionary biology and ecology, medicine and physiology, and the intersection of science and public policy. Jennifer West is the Fitzpatrick Family University Professor of Engineering at Duke University. Dr. West is a world leader in both nanotechnology and bioengineering. For example, she uses gold nanoparticles to detect and treat cancer cells, and also develops approaches to the synthesis of biomaterials and to tissue engineering for regenerative medicine. We hope you enjoy this year's presentation by Jennifer West, entitled Nanotechnology and Biomedical Engineering.
Thank you very much. It's really an honor to be invited back. It's certainly an honor to be here anytime, but to be chosen to come back for a second visit is truly a special honor. So today I want to tell you a bit about the work going on in our lab. And when we look at it, we're building really upon all of the things that have come before us in biomedical engineering. And already, if we look at medicine, there are many very important innovations that have come through biomedical engineering that are impacting patients today. We have a wide variety of different orthopedic implants. We have heart pacemakers, left ventricular assist devices, vascular grafts, a wide variety of prosthetics. And when we look at these, most of them are mechanical and electrical devices. They're generally macroscopic, so they're large devices that you can see with your eye. They're usually designed to be permanent implants in the patient, or at least very long term. And when we look at why failures occur in these devices, it usually has to do with the way that the body recognizes these foreign materials that we've put into the body. So usually you have poor interactions between the body, the cells in the body, proteins in the body, and the materials that we're making these implants out of. And so if we can gain a better understanding of the biological interactions when we put materials into the body, if we can start thinking about, instead of having permanent implants, having very temporary implants that are there to guide some sort of regenerative or restorative process of the body's own doing, so that the implant's not needed long term, then that can potentially get around some of these issues. And also, as we start to have a better understanding of the biology and better control over materials, we can start to make microscopic and nanoscopic devices as well. So when we talk about microscopic or nanoscopic, we're talking about the size of our materials and devices. And so here what we're looking at are different size scales, and then things that occur in biology and things that are man-made. So when we look at the size of a cell, for instance, here we see a picture of a red blood cell on the left. You're typically looking at something that you can measure in microns. So that's 10 to the minus 6 meters. It's 1,000th of a millimeter. And so when we look at some of the man-made technologies that work well there, a lot of the work in the electronics industry has really given us some fabulous tools that work well for controlling materials and their properties at the microscale. So we can use a lot of the tools from things like photolithography to help us make materials where we control their properties or their total dimension at this scale that's relevant to a cell. If we go down 3 orders of magnitude, we're then at a nanometer. And we start measuring things in nanometers when we're talking about individual proteins, DNA. These are things that are going to be nanoscopic. And so if we want to be working at this subcellular size, we really need to start being able to control materials at this size scale. And the past several decades have really given us some unique tools and abilities to work there. And some of the earliest work that really led the way for this nanotechnology revolution actually came from Sir Harry Kroto, who you heard from, as well as some former colleagues of mine at Rice University, who found the buckyball, which you see in the bottom right here, where that's a man-made material where you now have this exquisite control over size at the nanometer scale. So if we start looking now at trying to solve biological problems at these different size scales, I want to give you an example of how controlling materials at a microscale can allow us to change biological responses, and then how we can control materials at a nanoscale to enable other types of applications. So if we start off at the microscale, the scale of our cells, if we think about cells growing in our tissues, they grow in a substance called the extracellular matrix, which is going to be a network of proteins and polysaccharides that provide support for the cells, both in terms of mechanical support to allow them to form into three-dimensional tissues, but also they provide a lot of signaling support. They give the cells the cues they need for survival, for growth, for differentiation, and it's not just a matter of what signals are provided to the cells, but also how they're spatially arranged. So in this diagram here, you can see that usually you'll have clustering of different types of signals into microscale patches, and the size and arrangement of those patches can have a very powerful influence over how the cells will respond to the different signals. So we need materials that allow us to control what types of interactions occur and also how they're spatially arranged. So a material that we've worked with a lot is something called a hydrogel. So this will be a very soft polymer material that has a lot of water in it. So that makes it very compatible with cells. It will have mechanical properties that you can tune to match many soft tissues by changing some of the hydrogel properties. So it has a lot of the right kind of mechanics and chemical properties for use with cells and tissues. The base of the material, and in this diagram it's the black lines, is a very bioinert and safe synthetic polymer called polyethylene glycol. And we have that to be a background material that gives us essentially no biological interactions. And that allows us then to have a blank slate upon which we can engineer the specific biological interactions that we do want to have occurring. And so we can tether in different ligands that will bind to receptor proteins on cells and allow specific cell material interactions. We also design these materials to be biodegradable, but we try and take advantage of the mechanisms that cells would use to degrade extracellular matrix in processes like wound healing and embryogenesis. And for this, they don't just have their extracellular matrix degrade randomly by interaction with water. They instead have specific enzymes that will break down the extracellular matrix around them in a very specific manner and with a high degree of control. And we can take understanding of these processes and build them then into synthetic materials. And here in the yellow, we show putting in short substrates for these enzymes into the backbone of our hydrogel network so that as cells are growing in the material, they can use their enzymatic processes to degrade the material and create room for new tissue formation. So we also want, though, I mentioned, to be able to control where and how we're presenting different signals to cells. So we want to be able to create very controlled microenvironments within these materials. So with all of the different polymers that make up this hydrogel, at their end groups, we have CC double bonds, these acrylate groups, and these are very reactive and will react with one another and cause covalent bonds to form and link the polymers together into the hydrogel network. And we can either initiate this chemical reaction process with a chemical initiator kind of uniformly through the material, or else we can use something called a photoinitiator. And a photoinitiator is just a substance that will only start the chemical reaction when it interacts with a certain wavelength of light. And so by moving this to a photochemical process, we then have a lot of tools that have been developed for photolithography that we can incorporate to allow us to have microscale control. So this is an example looking at a cell grown on the surface of a hydrogel where we were presenting patterns, which are kind of the orangish-red dots you see. The cell has its actin fibers stained in green, and you can see that the cell and its actin fibers have aligned in the direction of the patterned ellipses. So just how we presented this to the cell had a profound influence on then how the cell organized and interacted with this material. So these patterns were formed by using highly focused laser light to control where we allowed photochemistry to occur when we were cross-linking in the ligands that would bind to the cell surface receptors. And so we can then use this process to make things besides shapes that we can randomly draw. So, we can go into any drawing software and we can make ellipses and squares and circles and things. But when you really look at cells in tissue, those aren't the kind of shapes they're interacting with. So what we decided to do was to develop an image-guided biomimetic patterning process where we could take an image of a cell, isolate out just the parts of the image that relate to specific biological structures we were interested in, convert those into scanning instructions for the laser to tell the laser kind of when to turn its shutter open and closed as it goes across the material. And then we end up cross-linking in the adhesive ligands for the cells just in those regions where we expose to the laser light. When we do this, we then end up with a pattern now that the cells can interact with, where instead of being random geometry, we're looking at whether or not we can guide new cells to adopt the same type of phenotype as the original cell of interest. And so one of the things that we've done with this is to make very large arrays. So at the bottom of this slide, you see an array where you're looking at many, many cells that now look essentially all identical to each other because of the pattern that they're growing on. And so the benefit there is now we have cells that are much more uniform in the way that they're getting their signals and thus the way they should respond. So if we want to be screening new drugs or understanding the role of a cytoskeletal protein in a certain response, here we can start with a much more homogeneous population. We can change the pattern so that we change the cell phenotype and look at a different array of cells and again get a much more homogeneous response and start to understand that. So one of the other things we can do with this type of patterning is use it to very intentionally guide cell responses. And so one of the things we were interested in was differentiation of a class of bone marrow cells called mesenchymal stem cells. And so these cells are in everyone's bone marrow and have the ability to differentiate into many different types of cell lineages in your body and can help participate in things like repair of injury. So there had been some early work looking from a lab at University of Pennsylvania looking at how mechanics can help guide differentiation of these cells. And really, the mechanics in the cell comes largely down to how they're interacting with the material around them and how their adhesive interactions are relating to the cytoskeleton of the cell. So we wanted to see if we could accomplish a similar control over stem cell differentiation just by how we presented adhesive ligands. So what we chose to do was to look at the adhesion patterns in differentiated cell types. So we were comparing in the top adipocytes, which are fat cells, to osteoblasts, which are bone-forming cells on the bottom. And we see some striking differences in the way that the adhesive patterns look, the size of the adhesive domains, how uniformly they're spread across the surface of the cell versus clustered into regions. And so we took examples of each type of cell and made patterns of them and then seeded mesenchymal stem cells onto them and looked at what ended up happening to their differentiation. And what we ended up finding was that if we seeded stem cells onto the pattern from adipocytes, that the vast majority of cells differentiated down a fat lineage. So here we're looking at staining with a red dye that highlights where you have lipid droplets within the cell. And so on the left, we can see this cell that's forming these very large lipid globules. On the right, we have a cell on an osteoblast pattern, and we see from its staining for a marker called RUNX2 that it's going into a bone differentiation pathway. And so we can now start to use this approach to control what cells do and how they behave. So if we want to look at taking these types of technologies and applying them to actually build new tissues, we need to be working in 3 dimensions. So what I've shown so far were patterns on surfaces, but we really need to construct 3-dimensional objects to end up with something that could be an implantable tissue. And so the approach that has been used for this field, which is referred to as tissue engineering, is to have some sort of polymer material that's referred to as a scaffold. And so we can use our hydrogel material as a scaffold. You seed it with cells, and because the scaffold is 3-dimensional, it allows the cells to grow in 3 dimensions, start to secrete their own extracellular matrix, which ultimately replaces the scaffold and gives you something that's very similar to the tissue. So this has been successfully done for a few tissues. So it's been successfully accomplished in skin, in cartilage that's non-load bearing, so people haven't made really strong cartilage yet, and in bladder. And what those tissues have in common is that they're either very thin or they're avascular. So one of the requirements to make a real tissue, so something that is larger, thicker, and unlike cartilage, which is very unique for its complete lack of blood vessels, would require a blood supply, that you have to figure out how to grow capillaries throughout this structure as well as your main tissue cell type. And that's been one of the most significant roadblocks and why we're not seeing things like tissue-engineered liver and things like that available yet. So that's been one of our focuses, is how we can get these capillaries and other blood vessels to form in our scaffolds. So here we have a 3-dimensional scaffold where we seeded into it endothelial cells that are stained green for the movie that's playing on the left. And what you're looking at is the first 24 hours of culture of those cells. And so they go from being spontaneously distributed throughout the material to organizing themselves into a microvascular network where they're forming tubules that interconnect and end up forming functional capillaries. So on the right, the still image is a picture of these microvessels in an engineered tissue that includes hepatocytes to make a tissue-engineered liver, so they're in red. And we can see that we have these functional vessels with the hepatocytes growing in between them. So those results are in materials where we have the right ligands there for the cells, the right supportive environment, but where we haven't done any patterning. And patterning in 3 dimensions ends up being quite a bit more difficult than doing it on a 2-dimensional surface. And so there we really had to create a new technology that would allow us to accomplish this. And so we developed something called multi-photon laser scanning lithography. And with our photoinitiators, they need to go to an excited state before they can make chemistry happen. There's a process called multi-photon microscopy that a lot more people are familiar with, and it's a very similar process to what we see in excitation of fluorophores from multi-photon microscopy. So if we're talking about a fluorophore for microscopy, you go to an excited state, and in the relaxation phase, you get fluorescence. You get an emission of a new color of light. And you can get there by interacting with 1 photon, which would be the standard practice, or you can go to a longer wavelength, so lower energy photons, and get there in multiple jumps. And so if you're doing 2 photon excitation and you've doubled the wavelength, you get there in 2 jumps. Those 2 photons have to hit in very rapid succession so that you don't have any relaxation in between them and that you can get the 2-jump effect. The likelihood of having that happen scales as the intensity of light squared. And the benefit of that is you can set up your illumination and laser focusing conditions such that you only reach light levels high enough to excite 2 photon events in a very small spot in space right where your laser is the very most focused. And so now if this is our photoinitiator instead of a fluorophore, instead of having light emitted, you start a chemical reaction. So now we can think of this little dot in space right at the focal point of our laser as being a little spot where we have covalent chemistry occurring. And so we can scan that around in space, we can open and close laser shutters, and now have a great deal of control over 3D patterning. And similar to what we did on the 2-dimensional surfaces, we wanted to bring in biologically relevant patterns rather than just random shapes that we had been drawing. And so we developed a process where we could take images of tissue, and in this example here, we're looking at images of capillaries from a tissue slice, and we select whatever area that looks good. We can take a 3-dimensional image using something called confocal microscopy, and then we can convert all of the structures in that 3-dimensional image into scanning instructions again for the laser, now using this 2-photon effect so that we can scan in 3 dimensions and create a very complex free-form 3-dimensional pattern in our gel material, which we see in the green stain. And in the last panel F, we're comparing the pattern gel back to the original tissue and see extremely high fidelity between the two. And then if we look at how it impacts cells, if we pattern a gel that has cells seated throughout it, we see that within several hours of initial seeding in the top 2 images, that the cells assemble into structures that are starting to follow where we've patterned the instructive ligands for them. And in the bottom, we're looking at a series of images where we have in red the tissue, in green the gel, and then the last image we have cells growing there that are stained for CD31 that tells us where the endothelial cells forming the capillaries are. And again, that image is taken at 30 days, so even for long time points out, can really control those cells and hold them into a precise 3-dimensional structure. So we can do a lot at the micro scale, and pretty much just have given you a very brief introduction to those concepts. There are a lot of people around the country working on these types of things at the micro scale. If we go down again 1,000 orders in size, that brings us down to the nanoscale. And so in recent decades, there have been an enormous number of new types of nanoparticles that have been developed that bring interesting new properties that potentially can be useful in medicine. And a couple of examples of materials people have been looking at include the buckyballs and carbon nanotubes, semiconductor nanocrystals called quantum dots that give you this very bright and tunable fluorescence, and then a wide variety of different metal colloids which can give you different optical properties based on something called their plasmonic properties. And a lot of the hype around nanotechnology is the idea that we're going to make little robots that we can inject that can go in and snip your DNA and fix you up that way. And at this point, that's very far-fetched science fiction. The ability to control a dimension of a material at the nanoscale really remains the cutting edge, and I don't think we're anywhere near the point of having to worry or think about robots coursing through our bodies at the nanoscale. So if we're not going to make nanorobots, why is this still interesting? Is it still going to impact medicine? And there still are a lot of different properties that we get by having a nanoscale material. We can have very unique properties that emerge from a nanomaterial that you don't normally see with that material in a bulk phase. You can have unique biological access, where you start having things that are the same size as subcellular structures and really can start to access those areas of the cell. You can have very good carriers for drugs or imaging agents. You can have bioresponsive materials that can sense and respond to what's going on around them. And so a specific application we've looked at is the use of nanoparticles like this for cancer therapy, and taking a really different approach to cancer therapy where we're doing something called photothermal therapy. So what we want to have is a very safe and inert nanoparticle that is designed so that when it's injected intravenously, it will end up accumulating in tumor sites, largely due to the leaky vasculature. So in areas where you have tumor growth, you have to have very rapid growth of new blood vessels to keep up with the growth of the tumor. And these new blood vessels tend to be very poorly formed and will have gaps between the cells that are on the order of 400 to 800 nanometers in size. So if you can make nanoparticles, you can start to be able to escape through these gaps. You want to make them big enough, though, so that they're not escaping into normal tissues or being filtered through the kidney. So you need to understand kind of the different biological access points and design your particles to fit into kind of a sweet spot where they'll accumulate in the tumors. You can also design your particles so that they have recognition elements on their surface that will specifically bind to tumor markers or tumor vascular markers. So we can inject these particles into the bloodstream, allow them to circulate, accumulate at the tumor site. The particles by themselves are very inert and safe, but when they interact with near-infrared light, they absorb this light, causing rapid heating and destruction of the tumor cells through heat. The light by itself is chosen to be in a wavelength where the light is very safe to normal tissue. It will be harmless there. And it's only when you bring the two together, the particles and the light, that you'll have a therapeutic effect. So the particles you see here, they look basically like nanoscale malted milk balls. You have a core material that, for the work I'll show today, is silica, and then a metal coating that's gold. And by choosing how we design these materials very carefully, we get very tunable optical properties. So we can change the size and composition of each layer and change kind of where the particles interact effectively with light, and also whether or not we have mainly absorption or scattering. So this is an example looking at 4 batches of nanoshells that all had the same size silica cores, but where we had different thicknesses of gold shell on the outside. And so you can see as we go from a 20 nanometer thick shell down to a 5 nanometer thick shell, we're moving where this peak for absorption of light is occurring. And sometimes it's easier to look at samples, so this is a similar set of samples where as you go from left to right, you're getting progressively thinner shells. You can see that we're tuning the color of the particles. So I mentioned we can also control whether at that peak we mainly have absorption or scattering. And so for the photothermal therapy, we want to have most of the light be absorbed so that it's converted to heat and can damage the cells. We can also design the particles so that they'll mainly scatter light instead, and that will send light back out of your system and allow you to do things like imaging or different diagnostic tests with the particles. So we're choosing this near-infrared light because of the fact that tissue in a small region of the electromagnetic spectrum is virtually transparent. So between about 700 and 900 nanometers, you end up above the absorption of chromophores like hemoglobin and melanin, but below where water starts to be a significant absorber. And so if you design your particles to have orders of magnitude stronger absorption than the tissue, you can set up your illumination conditions so that you can have very safe light for the normal tissue, but induce rapid damage when the light interacts with the particles. So for a cancer therapy application, then the idea again, to have these be very strongly absorbing in the infrared. If we want to, we can have targeting agents on the surface of the particles. We inject them intravenously, allow them to find the tumor site, and then we can apply light from outside the body and hopefully kill the targeted cell. In an animal study, the way it would work would be to have a mouse that has a tumor grown on it. You inject the particles intravenously. In areas where you have tumor growth, you'll have these leaky blood vessels, allow the particles to escape there. In the healthy tissues, the blood vessels won't have any of these gaps, and the particles stay in the bloodstream. So that allows the particles to accumulate at the tumor site, and then we can shine the near-infrared light from outside the body. So this is using MR thermal imaging to look at heating of two tumors in the back of a mouse, and everywhere that turns blue, we had a change in temperature of 15 degrees C, which is sufficient to cause irreversible damage to the tumor tissue. So the normal tissue in between the two tumors did not heat up at all and wasn't affected. And so we can take those animals and look at what then happens to them over time. So we know the thermal profiles we achieve in their tumors. We can look at how well it ends up affecting their therapy. And so in this study, we have 3 groups of mice. In green, groups that were treated with nanoshells and the laser, red, the laser alone, and blue was completely untreated animals. And at the day of treatment, they all had very similar sized tumors, but 10 days later, the 2 control groups had had very rapid tumor growth, whereas in the nanoshell treated group, we saw no detectable tumor tissue left in any of the animals. And this then went on to match up to their long-term survival, where the 2 control groups died due to excessive tumor growth less than 3 weeks after treatment, whereas the nanoshell-treated animals survived the duration of the study with no tumor regrowth observed. And so this and a number of other studies allowed the technology to move from the research lab and animal studies into human clinical trials. And to do this, we had to form a company, Nanospectra Biosciences, to have the manufacturing facilities, to run all the FDA-required types of testing. And so working with Nanospectra, this technology's been able to move into 3 different human clinical trials, one for recurrent refractive head and neck tumors, and then more recently, prostate and lung. And so the bottom picture here is a picture of a person's prostate. So the first group of patients treated in the prostate study were patients who were scheduled for radical prostatectomy. And we were able to do the photothermal therapy process just before their prostatectomy. And that gave a very unique opportunity to get human tissue back and to be able to really assess what had happened with the therapeutic approach. So we were able to see very good thermal damage of the tumors and little to no damage to the surrounding normal tissue. So we're continuing to look at advancing this technology and developing the 2nd and 3rd generations that will come out. So the very first generation that's in human clinical trials right now, the particles get to the tumors just through the leakiness of the blood vessels. And that was actually important for a first step into humans because that left our product regulated by the FDA as a device. Whereas when we add targeting agents onto the particle, the regulatory process becomes a little bit more complicated, and so we wanted to go through without that first. However, because we can very easily attach things like antibodies, peptides, or aptamers onto the surface of these particles, we have the opportunity to have very specific recognition of markers on tumor cells or tumor vasculature. And so in this EM image, you see a nanoshell, which is the black large object in the center, and around the outside, we have antibodies attached that have gold colloid tags to allow visualization by electron microscopy so that we can see the halo of antibodies bound to the nanoshell. And so if we look at the degree of specificity that this can allow us, here we have a co-culture between breast tumor cells that are on the top half of the image and normal fibroblasts, which are just skin cells, on the bottom half. And so at higher magnification to the right, you can see the dark staining where the nanoshells are present on the targeted tumor cells, but the immediately adjacent fibroblasts at the bottom show little to no binding of particles. And then if we expose those cells to an infrared laser, we see that we're able to very effectively kill the breast carcinoma cells that are above the interface, whereas the fibroblasts below the interface were unharmed by the process, and even fibroblasts that were immediately adjacent to a breast tumor cell were able to survive this process due to the specificity of the nanoparticle targeting and resultant heating. So we can target all sorts of different tumors just by choosing appropriate molecules that would be expressed. This is an example looking at targeting glioma cells. And we think that brain cancer will be a place where targeting is particularly important, since you frequently will have very close interdigitation between tumor cells and normal neurons that may be critical for function. So this is looking at a glioma study where we can see 2 tumor sites, and again using photothermal imaging. And you can see that we're able to get sufficient nanoshell accumulation so that we can accomplish this photothermal heating very effectively. So I've mentioned before, we can have our particles designed for absorption or for scattering for imaging. We've also looked at designing particles to be somewhere between these 2 extremes and to allow us to integrate imaging and therapy onto the same nanoparticle platform. So this is some work that was actually done by Rebecca Drezik's lab at Rice University, looking at the use of nanoshells for imaging. And so what you see is in the last condition here, which is the only one that's not a control, that you can see all of these tumor cells very nicely. We can see each individual tumor cell, they light up beautifully, and that's just due to the fact that the nanoparticles bound to their surface are scattering the infrared light very strongly. So in designing a system for integrated imaging and therapy, we had particles that we designed to be 50% absorbing and 50% scattering so that we could hopefully accomplish both goals. We injected them into the mouse with a tumor, allowed them to accumulate there, and then we first did imaging where we used something called optical coherence tomography. And on the bottom left, you can see we have a fiber optic probe that's just running along the skin of the animal and developing the image. And then we come back with higher intensity light to generate heating sufficient for treatment. And so these are what OCT images look like. They're somewhat similar to a B-mode ultrasound image, and you're looking at basically slices through the tissue. If we compare normal tissue with or without nanoshells, so on the left column is without nanoshells, the right column is with nanoshells, we see essentially no differences in those images. But if we look at the tumor tissue with and without nanoshells, we start to see enormous differences in the tissue. And when we do the image processing, we end up with signals that become much more effective for diagnosis of the tumor. And then if we look at survival of these animals over time, with these particles that are now only 50% absorbing, we still had 80% of the animals survive tumor-free long-term. And so we think that we still have something that is a very good cancer therapy, but also now gives us the imaging modalities as well. And so we're looking at whether or not we can extend this to multimodal imaging, where in addition to optical imaging, you could also accomplish CT imaging, which you can get because the electron density of the gold allows for X-ray contrast. And then we've developed versions of these particles that incorporate gadolinium, which is an MR contrast agent, so that we can do MR imaging as well. And so if we look at images just of materials that have these nanoparticles embedded in them, we see the top row are the gadolinium nanoshells, the next row are regular nanoshells, and the bottom is just a pure control. So we see that we only get the MRI contrast, the first column, when we had the gadolinium incorporated. We get nice X-ray contrast in both cases. And then we have 3 different modes of optical imaging: OCT, reflectance confocal, and 2-photon luminescence, that all give us very nice signals as well. The nice thing about merging these different imaging technologies together is that they give us different levels of magnification and different information about what's going on in the tumor. So MR and CT allow us to have whole body imaging. We can find where there are suspicious sites throughout a body. You might have metastases in unexpected locations. And so you can find these in your whole body images. The optical approaches give you much higher magnification images and a lot more information, but you could never scan the whole body looking for things that way. It would be like looking for a needle in a haystack. And so we can go from MR and CT to optical coherence tomography, which gives us resolution at tens of microns. And then we can go down to photon luminescence, which gives us resolution at the nanoscale, where the red dots there are individual nanoparticles that we can see through that process. And then we still have the ability to have a therapeutic effect. So the bottom panel shows changes in tumor size for animals from this study, where again the green is when you have the nanoshell treatment, and there we had 6 out of 7 animals have complete regression of their tumors. So we can also look at combining nanoshell therapy with drug delivery. So there's been a lot of work from other groups showing synergism between hyperthermia and different chemotherapeutic drugs. And so we can look at ways to incorporate site-specific chemotherapeutic delivery with this approach. And so what we've done is we've looked at putting thermally responsive polymer coatings onto the surface of individual nanoparticles. You can load these with a chemotherapeutic drug. When the particle heats up, it causes a phase change in the polymer, which causes expulsion of the drug from the polymer. So this is an example on the left looking at the amount of drug that's being released from the particle when the laser is off versus on in the gray panels. We see we can drastically change the rate at which drug is being released. And then when we look at the drug uptake into tumor cells, the drug is red in color, so we can actually image it and quantify it. We see much higher drug levels for the ones that had the exposure to light. So for the high school students in the audience, I wanted to just finish up with a couple of words. So hopefully you found this past couple of days all of this talk from scientists interesting. The theme of this meeting is where does science go from here? And really we go wherever the next generation is going to take us. And so it's important that we get the best and brightest students to pursue careers in science and engineering. And so when we think about what scientists really do, you've probably gathered that I do research, and then being at a university, I'm also involved in teaching. But when I look at what I really do, it's actually many other things. And so that's one of the great things about being a scientist, is it's a very dynamic lifestyle, usually where you're doing lots of different types of activities in different phases of your career, and you really have opportunities to pursue different interests and build a career that interests you the most. And so just in closing, I think that we're at a very exciting time between our knowledge of the Human Genome Project, advances in super resolution microscopies. We're getting more and more information about tissue structures and functions. And we can start to really translate these into new technologies to impact medical care. I think we're at a time where it's going to become increasingly exciting over the next decades. So hopefully the young people here today will want to join us and be part of the future. And this last image here was one that Rick Smalley, a former colleague, made. So he was one of the trio that shared the Nobel Prize with Sir Harry Kroto. And so when he would give talks to high school students, he always had this as his last slide. So in homage to him, be a scientist and save the world. So with that, we have more information here, and thank you.
So if you'll give us just a moment to swap technology out, we'll get the next speaker up shortly.